Today we're going to talk about Mycobacterium. It's a bacillus, and although it's considered gram-positive, many a times it may appear gram-negative depending upon which stain penetrates and reaches the cell wall. Penetrates what? A thick coat of a variety of lipids around the cell wall, which are unique to this genus, Mycobacterium, and some other filamentous bacteria. And the one lipid that is relevant to gram stain is mycolic acid. It is this thick layer of mycolic acid which renders the cell impermeable to gram stain. And due to this unpredictability in obtaining consistent results with a gram stain, Mycobacteria are often called gram indeterminate because you cannot determine for sure whether it's gram positive or gram negative. They are also called gram neutral or gram host. So to visualize this bacterium under the microscope, certain stains were developed that would not bind to the peptidoglycan as in a conventional gram stain, but would instead bind to the barrier that stops the stain from reaching the peptidoglycan, that is mycolic acid, and at the same time be resistant to decolorization, this time with acid alcohol, unlike acetone alcohol as was in gram stain. And this resistance, this property of resistance to decolorization is called acid fastness, and the bacilli are called acid fast bacilli, or AFB in short. Now before we discuss the various staining techniques for acid fast bacilli, let us touch up another important mycobacterial lipid, that is lipoarabinomannan or LAM. If we break this term, we get lipo, that is lipid, arabino, or because it contains the 5 carbon sugar arabinos, and mannose, it contains the 6 carbon sugar mannose. It's a virulence factor which inhibits the fusion of phagosome and lysosome. Actually, Mycobacterium resides and proliferates in a macrophage, which is a type of WBC. And lysosome, you might be knowing, it has an acidic environment which degrades dead organelles and foreign particles with its hydrolytic enzymes. Phagosome has its phagocytic property. So if this fusion is inhibited, lysosome and phagosome, they won't be able to function properly. LAM also confers resistance to antibiotics because, as with mycolic acid, lipoarabinomannan also forms a thick coat around the cell wall which the drugs or antibiotics are not able to diffuse through or penetrate to get inside the cell. This way, LAM acts as a virulence factor and confers the bacterium immunity to the immune system of the host. Now let's move to the acid-fast stains. So these are the various acid-fast stains. The first one was developed by German bacteriologists Franz Zeil and Friedrich Nielsen. Hence it's named Zeil-Nielsen stain or ZN stain in their honor. Next is Gabet stain, then Kenyon stain. And all have the basic sequence of steps as in the gram stain. That is, first the primary stain is applied, which binds to the barrier mycolic acid and not peptidoglycan as I mentioned earlier. Then it is decolorized with acid and alcohol as I mentioned earlier. But the bacilli would be resistant to decolorization, which is why they are called acid fast. I mentioned this earlier. So everything else in the background which does not have mycolic acid would be easily decolorized and ready to take up new stain. So when the counter stain is applied, the background would be stained blue by Loeffler's methylene blue. But the bacteria would be spared because methylene blue does not bind to mycolic acid, and anyways the mycolic acid is already saturated with the primary stain, so it cannot take up any more stain. Now let's discuss the primary stain, that is carbol fusion, which is composed of phenol, which is carbolic acid, and basic fusion. Phenol is the mordant over here, and heat enhances this action of phenol. Now as far as ZN stain is concerned, the primary stain is applied and kept for 5 minutes, during which it is heated. It is heated till fumes begin to emerge. These are the fumes of phenol, which is toxic and unhealthy. So to eliminate the need for heating, alternatives were developed, like the primary stain would be applied for a longer duration, like 10 minutes, as in Gabet stain, or the primary stain used would be of a higher concentration, as in Kenyon stain. And since there is no need of heating in these two methods, they are called cold methods. One thing to be noted over here, The counter stain used in Gabbett's technique is called Gabbett's methylene blue. Now, different concentrations of sulfuric acid used in the decolorizer have served different applications. Like 20% sulfuric acid helps us stain Mycobacterium tuberculosis, the causative agent of tuberculosis. When it is reduced to 5%, it stains Mycobacterium leprae, which causes leprosy. And when it is further reduced to 1%, as in quinine stain, it helps us visualize nocardia, the filamentous bacterium. And in all these techniques, the microscopic appearance is pinkish or reddish bacilli due to basic fusion in a blue background. And this is the characteristic appearance of an acid-fast bacillus. Now let's see a fluorescent stain called auramine-rhodamine stain. It's also called Truant's method. And the composition I have included at the bottom because I just missed it when I finished writing this line. So here the primary stain is auramine-rhodamine, a mixture of these two dyes, fluorescent dyes. The decolorizer is 0.5 to 1% hydrochloric acid dissolved in 70% ethanol. And the counter stain is potassium permanganate. And the appearance is greenish bacilli in a black or dark background. These are some other modifications of Zn stain. As we saw earlier, reduced concentrations of sulfuric acid in the decolorizer widens the scope of Zn stain. Similarly here, 0.25 to 0.5% sulfuric acid in the decolorizer helps us stain oocysts of Cryptosporidium, Microsporidium, Cyclospora and Isospora. These are all unicellular parasites. And Isospora is now called Cystoisospora. Donner stain, Moeller stain and Schaefer-Fulton stain, three different stains, are used for endospores. Donner stain uses 3% hydrochloric acid in the decolorizer and nigrosin as the counter stain. Nigrosin and India ink are two commonly used negative stains used to visualize the capsule, but they don't actually stain the capsule itself, but rather the background. So the background appears black and the capsule remains unstained. Ellis and Zabrowanny stain also uses 3% hydrochloric acid, but uses no phenol. In Muller-Chernoff-Turchitol method, there is no heating involved, where it becomes an exception. In all these techniques, the primary stain is heated, which is not the case with Muller-Chernoff-Turchitol method. And Turchitol is another alcoholic compound just like phenol. Phyte-Wade and Phyte-Farraco stains are used to visualize Mycobacterium leprae in histopathological sections or simply histological sections or tissue sections. Ellis and Zabrowanny stain is also used for tissue sections, and apart from these, several other modifications have been suggested by various researchers studying acid-fast bacilli, including the World Health Organization, but these are the ones most commonly used. And one last bit of information before we end the stain section. The Schaefer-Fulton stain that I mentioned over here is also called Wirtz-Conklin stain. We shall now discuss the various culture media for Mycobacteria and the relevant biochemical reactions. The most commonly used solid media is Lowenstein-Jensen medium, named after the people who first formulated it and then modified it. It's an egg-based medium. Other egg-based media are Dorset's medium, Petragnani's medium, American Thoracic Society medium, etc. Loeffler's serum slope, which is commonly used for Corynebacterium, may also be used for Mycobacterium. There are many other media, like Tarski's medium, which contains blood, and Pawlowski's medium, which contains potato. Loeffler's serum slope, as the name suggests, it has a slope or a slant. LJ medium also has a slant. They both are kept and prepared in McCartney bottle, and since they contain serum or egg, they are not sterilized by an autoclave. They are sterilized by a fractional sterilization technique called inspisation. Now, to inhibit the growth of contaminants, a dye called malachite green is added to these culture media, and Petragnani medium has the highest concentration of malachite green. Mycobacterium tuberculosis is an obligate aerobe, and 5 to 10% carbon dioxide facilitates its growth. The average generation time or doubling time, that is, the time it takes for a parent bacterium to completely divide into two daughter cells, is 20 minutes with E. coli as the model, Escherichia coli as the model. However, for Mycobacterium tuberculosis, it is 20 hours, and for M. lepri, it is 20 days. This is due to the slow metabolism of these organisms. MTB colonies on LJ medium are described as rough, tough, and buff. Rough because they are dry, tough because they firmly stick to the surface of the medium and they are difficult to lift or separate from the medium. Buff because they are buff colored or pale colored. They are also described as eugonic because the growth is luxuriant. Now the problem with culturing M. lepri is because it does not grow in artificial media. So, mouse footpad and 9-banded armadillo are the best in vivo hosts for culturing Mycobacterium lepri. Dysgonic is another term that describes the sparse growth of M. bovis colonies. Bovis comes from bovine because it primarily infects cattle and less frequently humans. Growth is sparse because glycerol present in LJ medium inhibits the growth of M. bovis, but it enhances the growth of MTB. That's why MTB had an eugonic or luxuriant growth. But if LJ medium is supplemented with pyruvate, it enhances the growth of both M. bovis and MTB. So a medium where pyruvate has replaced glycerol provides the optimum conditions for the growth of M. bovis. And this purpose is served by stone brinks medium, which is again an egg-based medium. M. bovis, whenever it infects humans, most often causes extrapulmonary TB, and extrapulmonary TB is commonly the reason behind fever of unknown origin or pyrexia of unknown origin. Another point of difference between M. bovis and MTB is M. bovis is microaerophilic, that is, it survives in low concentrations of oxygen, whereas MTB was obligate aerobe. Middlebrook 7H10 is another solid medium whose composition can be summarized as OADC, where O stands for oleic acid, A is for albumin or bovine serum albumin BSA, D is for dextrose, and C for catalase. Middlebrook 7H11 is same as Middlebrook 7H10 with the addition of casein hydrolysate or pancreatic digest of casein. Middlebrook 7H9 is also similar to Middlebrook 7H10 but without agar. So it's a liquid medium and it's supplemented with Panta mixture of antibiotics to inhibit the growth of contaminants. P stands for polymyxin D, A is for amphotericin, N for nalidixic acid, T for trimethoprim, and A for azlocillin. Middlebrook 7H9 is a fluorescent medium and it's used with an automated technique called MJIT or Mycobacterial Growth Indicated Tube. GeneXpert and CBNAT are two other examples of applications of automation in diagnosis and research. IgRA measures the T cell release of interferon gamma because actually Mycobacterium infection elicits a delayed type hypersensitivity response or a type 4 hypersensitivity response, which is mediated by T cells. Luciferase assay is used to detect drug resistance. Luciferase is an enzyme which is obtained from fireflies and in the presence of ATP produces light. So if the bacterium is resistant to a particular drug, it would produce normal amounts of ATP and normal light would be produced. But if it is not resistant, the quantity of ATP it produces would be low and consequently light produced would be low. And finally, Grafts medium and Mycobactocell are two other media that are supplemented with antibiotics to inhibit the growth of contaminants. We shall now discuss the various biochemical reactions but only in brief because they are not used in routine diagnosis but mainly in research because these tests differentiate between mycobacterial species that don't usually cause disease in humans and are found in the environment as saprophytes. These atypical mycobacteria are also called non-tuberculous mycobacteria or mycobacteria other than tuberculosis, and they were classified by Runyon and Timpe. They observed that some of them are slow growers and some are rapid growers. We saw previously that the typical mycobacteria take at least 2 to 3 weeks to develop observable colonies. But here, even the slow growers grow faster than the typical bacilli, and the rapid growers take less than 7 days. They also observed that some of these bacteria produce pigment on exposure to light, so they named them photochromogens. Photo means light, chromo means color, gen means to generate, while others produce pigment even in the absence of light. So they were named scotochromogens. Scoto means dark, but it's not that they produce pigment only in darkness. And non-chromogens do not produce any pigment. And some of the rapid growers are late pigment producers. Now these are the parameters or biochemical tests on which the atypical mycobacteria are differentiated, or even MTB and M. bovis may be differentiated. They may be high catalase if they produce a column of foam or bubbles which measures greater than 45 millimeters. And if it's less than 45 millimeters, they would be low catalase. Now whether they hydrolyze 2-in-80 or not, 2-in-80 is a surfactant, and whether they reduce nitrate or not also differentiates. Some of them tend to be slow growers at a particular temperature and a rapid grower at another, and some of them are a photochromogen at one temperature and a scotochromogen at another. Niacin, which is vitamin B3, is a metabolic product of all mycobacteria, but if they cannot process it or catabolize it further, it would tend to accumulate in the medium, and egg-based media are recommended for this test. Then there is an aryl sulfatase test, which has two variants, 2-week and 3-day variant, and the 3-day test may be carried out in a solid or a liquid medium. Whether they are sensitive or resistant to pyrazinamide also gives us a clue. Pyrazinamide is an anti-TB drug, and the detection of peroxidase and urease enzymes are also differentiating features. Before we start with the clinical presentation, we can gloss over some miscellaneous facts. All species of mycobacteria are non-motile except Mycobacterium marinum, which causes fish tank granuloma or swimming pool granuloma. And recent research suggests that even Mycobacterium smegmatis may also be motile. These are some other examples of bacteria that have varying amounts of mycolic acid around their cell wall and thereby exhibit varying degrees of acid fastness. Segnilliparas has an even higher content of mycolic acid than Mycobacteria. Then there is Nocardia, for which quinine stain is used, and Corynebacterium has the least amount, which causes diphtheria, so it's not acid fast. Cod factor is another virulence factor like lipoarabinomannan, which inhibits the fusion of phagosome and lysosome. It is composed of 2 units of mycolic acid, hence dimycolate, and trehalose, which is a non-reducing sugar. Trehalose is commonly found in fungi, and the prefix myco is used for anything related to fungi. And mycobacteria also have many features similar to fungi. But that's not the case with mycolic acid. Mycobacterial cell walls have many features common to both gram-positive and gram-negative cells, and their staining pattern depends on the phase of growth they are in. Hence, they are also called gram-variable. Now, there are some terms which we need to familiarize ourselves with before we can understand the clinical features. In a chronic inflammation, when the body cannot get rid of the irritant, macrophages fuse together to form a multinucleate condition called a giant cell. And in TB, these are called Langerhans giant cells. A multinucleate condition is also called a syncytium. These giant cells surround the foreign object and form a nodular lesion called a granuloma or a tubercle. And this is where the name tuberculosis comes from, because there is a proliferation of tubercles. These granuloma have a central necrotic area which has a cheese-like appearance. Hence they are named caseation, which is a hallmark of tuberculosis. We can now proceed to understand the clinical features of primary and secondary tuberculosis, and we shall do so in the form of eponyms. A caseation at the apex of the lung is known as Ghon's focus. And along with the Ghon's focus, if there is lymph node involvement, it's called a Ghon's complex. If a Ghon's complex heals with fibrosis and calcification of the lymph nodes near the hilum of the lung, it's known as Ranks complex. If the primary lesion originates outside the lung and then seeds or spreads to the apex of the lung through hematogenous route, it's known as Simon's focus. It may be reactivated to form Assmann's focus, which is an infraclavicular lesion. And a supraclavicular lesion is mainly called Poole's lesion, or may otherwise be known as Askoff-Poole reinfection. One striking feature of secondary tuberculosis, rarely seen in primary TB, is cavitation, which are air-filled spaces created due to destruction of lung tissue. It has a very high bacillary burden and is a hallmark of established infection. Miliary TB is a disseminated and fatal form of the disease, and it's named so because the tubercles resemble millet seeds. It also has eponymous features like Rich's focus, Wigger's focus, and Simmons focus, but we are not discussing them because it's a rare form of the disease and the video is getting lengthy. Rasmussen's aneurysm is another rare feature. I have included another infection because you might find it interesting. Mycobacterium avium intracellular complex consists of non-tuberculous mycobacteria that can infect the lungs. And when it affects elderly white women, it's called Lady Windermere syndrome, after Oscar Wilde's Victorian era play Lady Windermere's Fan, because people at that time believed it was an acceptable social etiquette for women to not spit, thereby predisposing them to lung infections. Avium actually comes from the class Apes because it primarily affects birds. And on a side note, MSC infections are particularly relevant for immunocompromised individuals like AIDS patients. Coming to the clinical features of leprosy, the two commonest ones are leonine facies, which means lion-like appearance, and saddle nose. Leprosy affects many nerves like the fifth cranial nerve or trigeminal nerve and the seventh cranial nerve or facial nerve. Other nerves that may be affected include the ulnar nerve, which causes claw hand, and the median nerve, which causes ape hand. If the lateral popliteal nerve is affected, it results in foot drop, and if the posterior tibial nerve is affected, it may lead to claw toes or hammer toes. And these are the alternative names of these conditions. Lastly, it's time to get acquainted with the terms that describe drug resistance patterns. Monoresistant describes the pathogen being resistant to one first-line drug. Drugs like isoniazid, rifampicin, and 3 others are first-line drugs. Pyrazinamide that we discussed in biochemical reactions is also a first-line drug. Polyresistant implies the pathogen is resistant to more than one first-line drug, one of which is either isoniazid or rifampicin, not both. Because if it is resistant to both isoniazid and rifampicin, it's called multi-drug resistant or MDR. In addition to being MDR, if it is resistant to either a fluoroquinolone or a second-line injectable drug, which are kanamycin, amikacin, and capreomycin, it's called pre-extensively drug resistant. And in addition to being MDR, if it is resistant to both a fluoroquinolone and a second-line injectable drug, which are these, it's called extensively drug resistant or XDR. And all hell breaks loose if it does not respond to any drug and is called totally drug resistant TDR or pan drug resistant PDR or extremely drug resistant XXDR. And such diabolical cases have been reported only in India, Iran, Italy, and South Africa. So this was all about Mycobacterium.